Hello everyone, my name is Kiera Cushway, and today I will be talking to you about my project involving the behavioral responses of an invasive fish species, Neogobius melanostomus, to the native freshwater mussel species Lampsilis siliquoidea. So first, a little background information. As many of you probably already know, aquatic invasive species are a major concern in Michigan and the Great Lakes region due to the introduction of as many as 182 non-native species in the last century and a half, most of which have been the result of international shipping traffic. Neogobius melanostomus, also known as the Round Goby, is an invasive fish species that was likely transported here in the early 1990s in the ballast water of ships, and has now invaded much of the Great Lakes region, wreaking havoc on native ecosystems, mainly through competition with other fish and predation on native fish eggs and fry. In addition to impacts on native fish species, Round Goby may also impact native freshwater mussel species, which are a highly diverse and imperiled fauna found throughout North America. Freshwater mussels are facing serious conservation concerns throughout their native ranges due to factors such as habitat loss and degradation, climate change, and the impacts of various invasive bivalve species like zebra or quagga mussels, or invasive fish species like Round Goby. Round Goby may impact freshwater mussels through more direct modes such as predation on smaller individuals, or by impacting reproduction, which I'll touch on in just a moment, or more indirectly through competition with native fish species that may be important to freshwater mussel reproduction as well. As I mentioned briefly earlier, freshwater mussels have a unique life cycle in which certain species of fish act as obligate hosts for the larval stage of the mussel life cycle, where larval mussels, also known as glochidia, attach to the gills or body of a host fish and remain attached for several weeks to months, until eventually dropping off into the substrate as juvenile mussels and continuing their life cycle. Freshwater mussel species often exhibit various host attraction strategies in order to successfully infect host fish, which can include the display of a lure that may resemble a prey item like a small fish or crayfish, as seen in this image here, or could take the form of a conglutinate or a package of glochidia, which may also resemble prey items meant to attract fish for glochidial infestation. Freshwater mussels may be host generalists or specialists, meaning that mussels may be able to infect and successfully metamorphose on the gills or bodies of many or just a few different fish species. This concept is important when we consider invasive species like Round Goby, as competition with native host fish species may result in reduced host fish populations that translate to reduced freshwater mussel reproduction success, especially for some of our more specialized mussel species. In addition, especially in areas where invasions are quite robust, it is important to consider whether freshwater mussels may be able to successfully infect and transform into juvenile mussels using invasive hosts like the Round Goby, a concept that has been tested and reported on in several studies, including one in the Great Lakes region. The other organism we chose for our study was Lampsilis siliquoidea, also known as the Fatmucket, which is a fairly common mussel with a broad distribution throughout Michigan. We chose Fatmucket as our study organism because of this commonness and distribution, and because they are a sexually dimorphic species, meaning that we were able to visually discern between male and female mussels. Fatmucket use lures as a means of attracting host fish and are more of a generalist species that are able to use a broader variety of hosts, including fish such as largemouth bass, walleye, and yellow perch. In addition, the Round Goby has already been identified as a potential host fish for Fatmucket in both laboratory and natural settings, although the success of glochidial transformation into juvenile mussels appears to be limited for mussels infecting Round Goby. Because of the negative impacts that invasive fish like the Round Goby can have on freshwater mussels, we decided to examine the behavioral response of Round Goby to the species Lampsilis siliquoidea in a controlled laboratory setting. Specifically, we wanted to examine how the Round Goby responded to the presence and absence of Fatmucket mussels, as well as any behavioral differences when interacting with male versus female Fatmucket. We predicted that there would be behavioral differences based on the presence and absence of Fatmucket and based on Fatmucket's sex, because of the reproductively important interactions between the species that may occur in nature. How did we do this? First, we collected mussels and fish from 2 sites in the Muskegon River in Michigan and brought them to the CMU vivarium. Round Goby were collected using hook and line technique, and mussels were collected using timed search surveys during July 2020. We also conducted preliminary surveys at the Goby collection site that indicated that no freshwater mussels were present there, although historically there have been 12 to 13 species found in the area. However, there were large amounts of zebra mussels present at this site, which was something that was notable. We gave all the animals about a week to acclimate to laboratory conditions, and then we began our experiments. Pictured here is the general laboratory setup we had, and what we would do is we would place a Fatmucket into a tank which was split in half using a plexiglass divider. Fatmucket were given approximately 1 hour to acclimate to the tank and assume a natural position, and then Round Goby were added on the other side of the divider and given an additional half an hour to acclimate. After this, the divider was removed and behaviors were recorded from 2 angles for approximately 1 hour. All tanks were also separated by impervious dividers, as seen to the right, to prevent goby in one tank from seeing goby in adjacent tanks. We ran a total of 10 trials, which each included 6 fish mussel encounters that involved either a male, female, or no Fatmucket mussel and a round goby. We then reviewed the recordings using a software called BORIS, which stands for Behavioral Observation Research Interactive Software. This program allowed us to set standardized behaviors that could be recorded with simple keystrokes, and also allowed us to review video at slower speeds if necessary to ensure that we were able to catch relevant behaviors that may have occurred relatively quickly. The software also recorded the moment that each keystroke occurred, so that we could go back and review recordings if necessary to compare multiple viewings and ensure that our analysis was reliable. Using BORIS, we recorded the percent of time that goby dedicated to behaviors such as movement toward or away from the mussel, moving up or down in the water column or remaining on the substrate, bumping or touching the mussel, burrowing in the sediment, or picking up or pushing rocks in the tank. Overall, we watched approximately 80 hours of video data, 58 of which were initial viewings, and then an additional 1/3 of the videos were re-watched to ensure accurate analysis. We then analyzed our results and compared the behaviors of round goby in the presence and absence of Fatmucket and in the presence of male versus female Fatmucket. Based on our analyses, we found that round goby did not significantly alter their behavior with changes in Fatmucket presence or sex. Generally, there were few discernible patterns of behavior arising in the different experimental groups, contrary to our hypotheses. These graphs show each round goby behavior on the x-axis in relation to female, male, and no Fatmucket, and the total percent of time spent on each behavior on the y-axis. And as you can see, there isn't a ton of variation in median values. Despite this, round goby tended to exhibit more varied behavioral responses when no mussel was present, although this wasn't the case for all behaviors. It may be important to note as well that there was quite a bit of variation in burrowing behavior, which stemmed from a few fish burrowing into the sediment for almost the entire encounter with Fatmucket, while others exhibited no burrowing behaviors at all. Throughout the course of the study, we had a single Fatmucket mussel that did exhibit luring behavior for a portion of the trial. This luring behavior only lasted for about 20% of the total interaction and ceased to occur after the Fatmucket was bumped by the round goby fairly early in the trial. The round goby did not exhibit any unusual behaviors during this trial that were outside of the range of behaviors exhibited in the presence of other female Fatmucket. In addition, there were few differences in horizontal and vertical movements of round goby based on fish size, visualized here with round goby size class on the x-axis and percent of time dedicated to movement toward or away from mussels on the y-axis, which was another parameter measured in our study. However, there was a tendency, as pictured on the right, for fish in the 12 to 14 centimeter size class to exhibit more upward movement than other size classes, a trend that was marginally statistically significant. Despite our lack of significant results, there were some notable encounters between Round Goby and Fatmucket that may warrant further investigation. In these encounters, individual Round Goby exhibited direct interactions with Fatmucket individuals that may have important ecological implications if they were to occur in a natural setting. During two encounters, a Round Goby pushed a Fatmucket with its rostrum, dislodging the mussel from the sediment and pushing it around along the substrate, as seen here. Similarly, here we again have a Round Goby and a Fatmucket, and the Round Goby dislodges the Fatmucket from the substrate and pushes it around on the substrate several times. As mentioned earlier, there was also one instance of luring exhibited by Fatmucket. This involved the female Fatmucket extending her mantle flap outside of her shell, where it moved in the water column and in a natural setting would serve to attract host fish. In this case, if you look very closely, you might be able to see the Fatmucket displaying her lure during the encounter, within the red circle. You also might not, but just know that it is in fact there. Eventually, the Round Goby disturbed her by bumping her with its tail, which caused her to cease luring for the remainder of the encounter. Despite our overall lack of significant results, there is some important knowledge to be gained here. Few studies have examined fish behavior towards freshwater mussels, except in the context of luring mussels with known host fish, so our methodology can provide a framework for similar behavioral studies that attempt to quantify fish behavior in relation to freshwater mussels. Our research can also inform management and conservation decisions with relation to invasive fish and the impacts that they can have on freshwater mussels. Specifically, while we didn't see any significant behavioral differences in Round Goby, the isolated interactions we captured could indicate that Round Goby may have isolated impacts on mussels in natural environments in a variety of ways. For example, interactions with luring mussels could result in premature release of glycidia or infestation of Round Goby as opposed to more traditional host fish, impacting reproductive success. In addition, dislodging mussels from the sediment could result in transport by strong currents downstream to less favorable habitats, as well as necessitating increased energy expenditure used for righting themselves after being dislodged. Our research opens up further questions regarding Round Goby behavior towards mussels, and there are several similar studies that may be interesting to pursue. For instance, it could be interesting to see how Round Goby respond to the presence of Dreissena polymorpha, the invasive zebra mussel, which is a known food resource for goby and which were present in large numbers at our fish collection site. Additionally, it could be interesting to further examine how Round Goby respond to luring mussels because we only had that one instance of luring during our experiment. Finally, it might be beneficial to examine how Round Goby that already coexist with freshwater mussels may respond to their presence. As I mentioned before, because mussel surveys at our fish collection site indicated that no native mussels were present there, although there have historically been found in the area. Exploration of these topics could help further clarify the interactions that occur between Round Goby and freshwater mussels in natural environments, lending to our ability to effectively manage these associations. In addition, we found that Boris, the software program used in our study, was an effective means of examining fish behavior, which could be widely applicable in fisheries settings and broader ecological settings as well, and allowed for review and comparison of data in ways that were both standardized and replicable, making it a valuable tool for similar studies. With that, I would like to thank everyone who has helped me make this project possible, including the researchers in Dr. Woolnow's lab for assistance with organism collection, Michael O'Neill in the CMU Vivarium for his knowledge and expertise in caring for animals in the laboratory, Dr. Kevin Pangle for providing cameras for recording, as well as Kelly and Karen Cushway for providing access to the mussel collection site, and the CMU Biology Department and Honors Program for providing funding. This project would not have been possible without the support from so many people who are willing to take the time to help me. If you have any questions or would like to learn more or access some relevant literature relating to our study, you can reach out to me at the email listed below or to my advisor, Dr. Dalen Woolnough. If you are also interested in the research our lab does and want to learn more or keep up with what we are doing, you can follow us on Twitter or Instagram for some fun and more informal updates on our research. With that, thanks for listening, and I hope you enjoy the rest of the conference.